Got it. I approve. So snowflake yeast are cool in that they have a nice little fractal growth form. I don't know if you can see my mouse. I assume you can. They have this cool sort of branchy tree. You can't? Okay, hold on. Let me go to laser pointer. They have a sort of branchy growth form, tree-like growth form. And I'll just replay this video here. You can watch them divide in real time. I guess this is not quite real time. It's a time lapse, but it have a nice sort of synchronous budding strategy. So daughter cells join the group from which their parents were in, making all the cells in these groups clonal. And then you get this kind of neat thing that as cells divide and they crowd their group, strain caused by cell-cell interactions loading up on these branches of cells actually results in giving them a life cycle for free. Like as soon as the strain accumulated from cells pressing on each other exceeds the tensile strength of a cell-cell connection, you break off a branch. And this is great for giving you a life cycle, which you need if groups are going to become Darwinian entities. They need to be able to make new groups. But it's actually not so good for building a tough multicellular organism. The material of a snowflake yeast is actually really bad. It's very brittle and very weak. You know, if you think about like the difference between a toothpick and a 2x4, they actually have the same materials properties, but usually as a material gets bigger, there are more bonds to break it, so it gets stronger. With snowflake yeast though, if you ever break a single cell-cell connection, it's like cutting a branch off a tree, the rest of that downstream branch floats away. So no matter how big it is, if you break a single cell-cell bond, you break the organism, which actually makes it a very, very weak material. So Ozan, was a postdoc in my lab, now he's a research scientist, has figured out how to do a really cool long-term evolution experiment. We're in the low 1,000, we're just past 1,000 transfers a few months ago. And so that puts us at we get about 5 generations per transfer. So about 5,000 generations for the experiment as a whole. And so he's been studying a number of things, but the thing I'll be telling you about today, we've mostly been looking at the role of oxygen and size and all sorts of cool stuff. I'll be telling you about macroscopic size. And so to do this work, we collaborated with Peter Juncker, who's a soft matter biophysicist here at Georgia Tech, as well as Eva Dyer, who's in biomedical engineering and is an image analysis guru, specifically scanning electron microscopy, as well as a bunch of students postdocs from our respective labs. And so if you look at what happens with respect to size over time, you see something pretty cool. At first, everything is evolving kind of, in lockstep. There's this parallelism where, there's 5 replicate populations in the treatment that eventually evolve large size, and they're essentially increasing in size at the same rate. And then 2 of them kind of peel off and get really big. We're talking 20,000 times the volume of their ancestor, whereas the rest sort of plateau, which is typical to what I've seen in these experiments. And I was excited to see something different, but they sort of plateau for a while. We're talking nearly a year of transferring and them not really doing that much before they eventually break through this size barrier, as it is, and then get large. Snowflake yeast are big and dense. So what you're looking at here are these, what we call macroscopic snowflake yeast. They're very macroscopic. They're substantially larger than a fruit fly. Here's the ancestor on the left and evolved on the right, 600 day evolved on the right. And then if we look inside the cluster, this is actually adding thin slices from serial bulk phase scanning electron microscopy to a sort of growing cube. You can look into the center of one of these things. You can see they're very densely packed. Now, the easiest way to get something which is 20,000 times bigger than the ancestor is just for them to evolve to become sticky and form aggregates of snowflakes. This shouldn't be hard in yeast. Yeast make all sorts of adhesive proteins on their cell walls to flocculate. But if they were to do this, we'd expect them to form chimeric groups. Here we've taken a single strain of macroscopic snowflake yeast and labeled it with either green or red fluorescent protein. And of 70 clusters that we looked at, 100% of them were monoclonal. They're either green or red. So they're not forming, aggregates. Although they do have this sort of globular dome. If you flatten them, which is what we did here, you can see them break down to these little modules, which look more like regular snowflakes. So what's going on? Well, we know a lot about the physics of these yeasts, and I'm kind of skipping over several physics papers we published with Peter's group in the last couple of years. But the first thing that snowflake yeasts do when they're adapting to get large is they actually evolve more elongate cells. And this does something kind of neat and a little counterintuitive. If they imagine building a sort of a tree-like growth growth form with ping pong balls, right? It gets dense very quickly, and it's hard to add new ping pong balls without this thing jamming. If you were to do the same thing, but with hot dogs, right, you'd make a much more spindly, much more sort of, it would take up a much larger three-dimensional volume, and there'd be a lot more open space around those cells. And that's actually helpful here because it prevents these branches from running out of space, jamming, and then causing fracture. And so what we typically see in these experiments, and here's another example of it, is that as you make the cells longer, aspect ratio is the ratio of length to width. As you make your cells longer, you get bigger groups. And so for the first, this kind of represents the first couple hundred days of evolution for different lineages. Everything is getting slightly bigger by getting longer cells at a rate that's kind of expected. And then you see this surprising discontinuity where all of a sudden getting longer cells gives you much, much larger groups. We don't really know how to explain this. If you look at sort of the expectation of how cell shape and length should affect the packing within the group, well, if we model this, we can model this with a three-dimensional biophysical simulation of snowflake yeast, then having more elongate cells should reduce the packing density or the packing fraction, the proportion of the volume that's biomass. It should reduce it because you're doing progressively more hot dog shaped cells. It should be more spindly. There should be more open space. And initially, our experimental snowflake yeast land right on top of this, which is cool to see because this isn't directly parameterized. So it's nice to see that we get similar packing fractions in our experiments and our simulations. But then we see this strange thing where as the cells get longer, all of a sudden, rather than continuing to get less dense, they're getting more and more dense. Again, something we can't explain. There must be something different about these macroscopic snowflake yeast that explains how they're bucking the trend here. So to do this, we looked at the topology and the geometry of these snowflake yeast sort of branches of cells. We tried all sorts of ways to image them with fluorescence microscopy, 2-photon light sheet. We tried everything we could get our hands on. None of it worked. Turns out that yeast cells are really bad for imaging with light because the cell walls scatter light and you're going in and out of cells frequently and it just diffracts light like crazy. So no matter what we did, we couldn't see more than about 40 microns into a cluster, which might be a millimeter in size. So instead, we brought them to a scanning electron microscope where we could take tens of thousands of thin slices and then image them. That's why this kind of looks like it was made in Minecraft, is that this is actually layers of hundreds of slices. Then we actually were able to then resolve the topology of our snowflakes. And we see, in fact, this entanglement, which we'd never seen before, where branch cells are actually wrapped around each other. And entanglement, this is a common thing in physics. It's a pretty easy thing to happen. And we know it can make materials very, very strong because to break something now, you have to break all of the connected entangled components. You're no longer breaking one bond, you're breaking thousands of bonds. It's a way to make something a lot stronger. So to see if our snowflake yeast really were entangled and if they were entangled enough to explain this, we borrowed a method out of polymer physics where we basically fit a convex hull. It's the minimum convex, anyway, it doesn't really matter, the technicality, but we fit these basically the bounding boxes around elements of branches of snowflakes from within these large clusters and asked if they overlapped with another branch. And if they are, then they're entangled. And then we can percolate that entanglement, starting with a single branch all the way throughout our cluster, or at least a subvolume of the cluster, and ask what proportion of the cells are mutually entangled. And the average was like 93%. Which is what you need actually if entanglement is going to provide enough, sort of coverage to prevent this big thing from falling apart. You need to have almost all the cells as part of the entangled component, and we do. What's more, we measured the materials properties of these macroscopic snowflake yeast by compressing them. We actually had to use totally different instruments to make the same measurements on the ancestor, which is tiny, and the macroscopic ones, which are big. We use an atomic force microscope for the ancestor and a giant 400-pound universal testing machine for the macroscopic ones. They're very different in scale, but there's this cool thing that comes from condensed matter physics and sort of also polymer physics, in which entangled materials have what's known as strain stiffening. So if you compress something which is entangled, the more you compress it, the more energy it takes to continue compressing it because those entangled connections begin to engage. And so we see that in our yeast. In the ancestor, they have a linear stress strain plot. There's no strain stiffening here. But in the macroscopic yeast, where they look like all, the cluster, the cells are entangled, we see this beautiful convex shape here, showing that as you begin to compress it, it gets harder and harder to compress, which is, again, strong indication that our cells are entangled. Now we can actually measure the toughness of these of these yeast by integrating the energy input to fracture and normalizing by the volume. The idea here is that a toothpick has the same toughness as a 2x4. It's just that a 2x4 is bigger, so it's stronger. Toughness normalizes out that change in volume. And even though we've normalized out the change in volume, the macroscopic snowflake yeast are 10,000 times tougher than their ancestor. And if you put this on a sort of toughness strength materials plot, we go from something which was in the ancestor a very, very weak material, where if you break one cellular bond, you break the group, right? It was 100 times weaker than gelatin, to macroscopic snowflake yeast, which are as tough and strong as wood. Like, they're actually a very strong and tough material, which is, it's really cool to, for me, this is one of these, like, I can't believe we can do this. This is super cool. It was neat to see how much material adaptation, right? The materials properties of the organism could evolve in just 3,000 generations. We've sequenced yeast from all 5 replicate populations, and I'm kind of going to breeze over this because honestly, I don't think it's really that insightful. We see mutations that largely make sense. We get a bunch of mutations in cell cycle processes and filamentous growth. If you look at all of these basically cause modifications to the cell cycle, so they spend more time in the phase of the cell cycle where you get essentially extension before you get symmetrical growth. It gives you longer cells. Prior work knocking out all these genes gives you longer cells for the most part. And we have changes in budding, which largely give us bigger bud scars. We think bigger bud scars gives you stronger cell-cell connections. So longer cells, stronger connections, it makes sense. We can actually mix some of these strengthening and lengthening mutations, and with just a few of them get clusters that are 100 times bigger than their ancestor. It's cool, but I don't think it really is that important. It's sort of just makes sense. But broadly speaking, I'm gonna wrap up here. I'm not sure how long I've been speaking. Oh, it's 4:14. Perfect. I'm gonna wrap up. I think physical processes play a very important role in the evolution of multicellularity. It sort of scaffolds this process of groups becoming Darwinian entities. Everyone here should be familiar with reproduction, heritable variation, selection, and adaptation being sort of a description of an evolutionary process. So here we have just one mutation, which gives us groups of cells from a unicellular ancestor. And those groups have physical strain problems, which give them a multicellular life cycle. It didn't evolve for a reason. You get it for free, but it allows groups to reproduce. That's very important. Now we can then play around with mutations that change the properties of individual cells within these groups. There's no developmental program or anything that sort of creates novel and heritable multicellular variation. We have a simple little multicellular critter here. It's just a dumb clump of cells. But if you have mutations which change the properties of individual cells, those actually have emergent properties because they're growing in a defined structured group. And so if you change their properties, you actually give rise to very predictable multicellular macroscale properties. We actually have two new papers on the bioarchive I haven't talked about, one from a quantitative genetics perspective, one from a biophysics perspective, which argues why, in fact, these emergent multicellular traits of cell level changes are more heritable than the underlying cell level changes themselves. So the emergence of novel heritable multicellular traits, that's easy. Selection can act on these. You got to cut me off?
Yeah.
All right. Just give me 30 seconds, okay?
Sure.
And sustained, adaptation. What we show in our LTEE is that we can see a lot of innovation occurring due to sustained adaptation, and understanding cell type specialization and differentiation and developmental programs. That's really where we're going right now with this system. I think that snowflake yeast are a great model system for a lot of the types of complex multicellularity we care about. And with that, I want to thank our group, our funders, our collaborators, and plug our graduate program in quantitative biosciences. If you think integrating this different types of perspectives and approaches is cool, let me know. Applications are due December 1st. Thanks.
Awesome talk.
Thanks.
John, do you want to, if John's here, do you want to set up your screen share?
Yeah.
All right, let me stop sharing here.
And then I'd encourage folks to ask questions in the Zoom chat, or the Slack chat is even better.
Oh, cool. I don't have Slack.
Oh, Zoom chat then, Zoom chat.
Okay, cool.
So let's see, are we, Zoom's in the way. Are you seeing my presentation or my presentation with the notes and things?
I see your notes and things.
Okay, so then I have to switch these. Okay.
Okay.
All right, good to go. Okay. All right, hi everyone, my name is John, and I'm a 5th-year PhD student in the lab of Premal Shah at Rutgers University, and today I'm going to talk to you about my thesis project, which involves characterizing the molecular phenotypes underlying 22 years of bacterial adaptation. Okay, so here's what the outline of the talk is going to look like. First, I'm just going to talk about like the goals of this study and the goals of my thesis in general. And then we're going to move on to answer some of these more specific questions given the data we've generated so far, but we'll hit those as we get to them. So overall, my thesis really revolves around understanding how molecular phenotypes affect evolutionary processes like adaptation and speciation and so on and so forth. And so the way we're going to study that is we're going to take a system like the LTEE, where we have these defined changes in fitness and the genome and all these kinds of observations about them. And we're going to study various molecular phenotypes. And when I say molecular phenotypes, what I'm talking about are transcription, which we're going to study by RNA-seq, translation, which we're going to study via riboseq, and then finally the metabolomes, and we're going to study that by mass spec. So we have 3 high throughput methods to study these genome-wide processes at the genome level. And so more specifically, what we're going to do is we're going to take the ancestral bacteria and then 12 evolved clones from the 50,000 generations, so the ones with the sequenced genomes. And we're going to, quantify these transcriptional, translational, and metabolic changes. And once we do that, we can start to ask other questions like how similar are the clones or how different are they? Among other questions about what exactly is different between the ancestors and the evolved lines. But this actually gets more complicated, right? Because the molecular traits we're looking at here can change minute to minute or even second to second, as opposed to the genome, which takes much longer to change. And we know that the bacteria in the LTEE experience these 2 distinct growth phases, which are exponential and stationary phase. So we can make these measurements in the exponential phase, but we can also make these measurements in the same measurements in the stationary phase. And once we have all of this information, we can start to look at growth phase dependent changes. And then we can also check to see if growth phase dependent changes from the ancestral bacteria are the same in the evolved bacteria. Are their evolved bacteria different among each other? There's a lot of different comparisons we can make here, and I intend to make all of them. So the first question that we can ask is that how similar or different are the transcriptional changes across the evolved lines? And this is going to come from our RNA-seq data. And so far, I only have my exponential phase RNA-seq data. So the first 3 bits will largely be from that data. But I do have metabolic panel data for the exponential and stationary phase, but just no sequencing data yet. So the first big observation we see is that there are some parallel transcriptional changes. So what you're looking at here is a heat map, and across the bottom there is 1,273 transcripts, and these transcripts come from a set of transcripts that have reached statistical significance in at least one of the evolved lines. So that means that when I calculated these fold changes with DE-seq2 from the ancestor to the evolved line, the transcript reached statistical significance in one of the lines. And so I'm considering this set as like the statistically significant set. And what I've done, just for visualization purposes on this heat map, is I've organized the genes from left to right in order of increasing mean fold change. And you can get this really striking red to blue gradient across the x-axis. And what that visually is indicating to you is that among the statistically significant set, there's a lot of genes that are downregulated in all the lines, and then there's a lot of genes that are upregulated in all the lines. And sometimes there's a mixture of those 2 properties, but for the most part, you really don't see so many blue bars on the left here, and you don't really see so many red bars on the right, though there are some. So overall, this is visually indicating to you that there's, appears to be a good amount of parallelism. But a reasonable question is, well, how many changes of these are actually shared? And that's what this graph here is showing you. So along the x-axis is the number of evolved lines that a gene has reached statistical significance and has a fold change in the same direction in n number of lines. So this, and the red bars are down regulations and the blue bars are up regulations. So this red bar is telling you that there is like 400 genes that have been down regulated in only one line. And you can see that this starts to taper off as we increase the amount of lines. So there are only a handful of genes that end up being significant and the same direction of fold change in a lot of lines. But the expectation isn't really so clear here. How many genes would you expect to see follow this pattern given all of the data we have? So what I did is, if you imagine this table where I have in each of the evolved lines, and then I have information on all of their genes, I have their fold changes, and I have their statistical significances. If I can shuffle those around within each line, it's essentially asking the question, and then redo the calculation that gives you this graph. It's essentially asking the question of, given the number of genes I observe to be statistically significant and having a fold change in a particular direction, how often would I expect to get a result like this? Like, how many times would I see 7 G, or however many genes this is, like, I don't know, 20 or so occurring, being downregulated and significant in 7 of the evolved lines, or 8 or 9 or 10 or 11. And when I do that a bunch of times, and I can plot the ratio of observed over expected, and I get this result. And it's really cool, because what it's saying is that, in fact, I observe less single or less changes in 1 or 2 lines than I would expect by chance, and I observe more changes in more numbers of evolved lines than chance. And in fact, through all the times I'd had that calculation redone, I think it was 100 or 1,000, I forget now. Never once did I have a gene or a set of genes by chance end up being statistically significant and altered in the same direction in 7, 8, 9, 10, or 11 lines. So that means that these genes that are experiencing alterations in the same direction in 7, 8, 9, 10, and 11 lines are probably truly exceptional, and that this isn't just some kind of random chance thing that we're seeing. So that's good news. Yeah, this table is still here for some reason. Anyway, so I've just given you a brief overview about transcriptional changes. And by the way, I will be keeping these overviews brief because we do have a paper on bioRxiv, and we should be putting out a revised one and hopefully a resubmission soon. So presumably you'll see a full-fledged publication in the near future, so you can read all about it there. But we've talked about transcription, but what about translational changes? So surprisingly, it seems that transcriptional changes are driving translational changes. What I'm showing you here is that we're observing a high correlation between our RNA-seq TPMs and our ribo-seq TPMs. So our, the abundances of RNAs and the abundances of ribosomes on those RNAs. And you might think this is not surprising, but in fact it is, because when you perform this kind of, when you look for this kind of relationship in other organisms, so what this graph is showing you is from a different paper, the relationship between RNA abundances and protein abundances, they're not really so correlated, and the correlations change over time depending on the organism or when the study was done. So it's not really clear cut what this, what the correlation between RNA abundances and protein abundances are supposed to be, which means that in a lot of organisms, especially higher level organisms and metazoans and things, there's a lot of translational regulation going on. But we don't really see that here for some reason. And we use this program called RiboRex, which, given a set of RNA-seq and ribo-seq data across 2 conditions, so namely the ancestor and the evolved strain, it can look for differences in the ribo-seq data that seem exceptional given the observed differences in the RNA-seq data. And when it comes down to is basically finding transcript-wide differences in ribosome density. And I'm showing you the number of genes which reach statistical significance in any one line, and it's very few. So somehow it seems that there's not so many global translational changes happening. But a neat thing about ribo-seq data is that you can assign each of the reads to a codon. And when we do that, I can give you this kind of heat map. And what I'm showing you here is the fold change in ribosome density at any one given codon from an ancestor to an evolved line. Red indicating that their ribosomes are spending or are less occupying of a particular codon, and blue indicating that they're occupying more of a particular codon. And that means that they're spending more time there, which generally means that for some reason translation could be slow at that spot. And a neat thing we see is that with the stop codons, there seems to be overall decreased density, which might suggest that translation termination may be faster in the evolved clones. I've not found a good explanation for this yet, but what that might lead to is faster ribosome recycling, and that could mean that more ribosomes are readily available to perform translation initiation, which is the rate limiting step of translation. So we've seen some expression changes, but like, what do they do? To figure that out, we did kinds of like Go and KEGG searches and things like that. And it turns out there's a good level of parallelism here, and it kind of goes up as we go down the hierarchical chain here, that there's a lot of metabolic pathways that are similarly altered across the clones. So here I'm just giving you an example of some of them. There's some KEGG categories that are reach statistical significance in the most number of lines, and you can see that, for instance, this top category here is the flagellar assembly, so having to do with the flagellar apparatus. And these are, this is a result that they had also found in the 2003 Cooper study of gene expression, which they did with radioactive microarrays, but that was at 20,000 generations. So it's interesting to see that we have patterns that may have reached fixation long ago still showing up in our data. Granted, the loss of the flagellar apparatus is a common adaptation in lab-grown E. coli because you don't really need to swim in the environment of a shaking flask, right? But now I gave you that slide at the beginning where I showed you we have all this data and we'd like to link it together in some way. And there's tons of other data from the LTE. So we'd really like to integrate all of those and link them together. So I'm going to give you an example involving NAD synthesis. So I don't know if you guys can see my mouse, but let's consider this gene NADR, which is the repressor to these NAD PNUC, NADB, and PNCB. This gene is already known to have fitness increasing mutations in the LTE. And if we look at the genomic data, I can see that every single one of the clones from our study does have some kind of mutation in it. Most of them are SNPs. One of them has an insertion element mutation in there. And then when I look at the expression data, I can see that the NADR gene itself is largely unaltered and never reaches statistical significance. And in this case, it has a gray box because it's had an insertion to it, which probably caused a frame shift, which probably destroyed the whole protein. So in my data, generally those end up as gray boxes on heat maps. But all of the genes with which it's supposed to repress have all reached some level of statistical significance, and they're all upregulated. And another neat thing you can see is that these mutations are kind of clustery, and it turns out that these, judging by where they're clustered, they do sometimes occur in DNA binding domains or other parts of the protein. So it very much seems that all of the evolved lines have had parallel inactivating mutations to this NADR gene, and it appears that the effect of that is upregulation of these genes. So then the most reasonable question is is there actually more NAD in the cells? Because this is part of the NAD synthesis pathway, and it's kind of obvious that having more NAD might allow you to grow faster and things like that. But this remains to be seen, right? Is there actually more NAD in the cells? And that's where our mass spec data comes in. So it does look like that there is more NAD in the cells. The mass spec data is quite new to me, so I've not really gotten around to doing a full analysis. But on the left here, there are heat maps from both exponential and stationary phase, giving you the rho Z score, so like the relative amount of whichever of these molecules in the cells. And on the right here are the ancestors. The orange ones are going to be your evolved lines that are non-mutators, and the red ones are going to be the mutators. And I think I forgot to mention that in all my graphs, when the lines are in red, they're the mutator lines. And for the most part, all of these, the ancestors tend to be these big red blocks, so they have the least amount of NAD, and most of the evolved lines have more NAD, except for these two, and sometimes. And you can see that the actual values coming out of the mass spec data, or at least the normalized ion counts on a log 10 transform in this graph. And you get the black points are again the ancestors, and you can see they're generally occupying the lower regions of the graph. So it very much seems like there is probably some more NAD in the evolved lines. when I was thinking of this, it didn't occur to me that the mass spec was going to grab NAD plus, NADH, NADP plus, and NADPH. I was just thinking like, yeah, NAD. I've never done like these huge mass spec studies before, so it was a surprise to me when I saw these in the data. And I'll have to think about, how the relative levels of each of them is going to matter when it comes down to all of this. But yeah, it's an interesting find for sure. And I'm hoping to make more of these kinds of connections. So as for conclusions, what I've shown you is that we're basically using multi-omics approaches to understand evolution at the molecular level and see how these molecular characteristics change over time and how they affect these processes. And that in the LTE, it appears that despite the different sets of mutations, the evolved clones experience similar transcriptional changes. And for some reason, that is what's driving translational changes as well. And that these, at least in the case of NAD, might appear to result in corresponding metabolic changes. But there's plenty of questions still. For instance, like, will parallelism remain during the stationary phase? The LTE bacteria spend most of their time in stationary phase. So you'd have to wonder if there was a place where they were going to show their differences, maybe it'd be there. How different is the evolved response to stationary phase compared to the ancestor? Experiencing stationary phase is not a new thing for E. coli. So I have to wonder how robust is the transition from exponential to stationary phase to all of this evolution that's been happening in the LTE? Does the switch from exponential to stationary in the evolved lines resemble the ancestor, or is there something fundamentally different about it?
Let's see.
And then obviously, what's the full extent of the metabolic?
I'm going to ask you to wrap up there so that we can
That's
move
it.
on to the next speaker. Great.
Yeah, I'll stop sharing.
Michael Travisano, do you want to set up your share and then we can do questions for that talk in the Zoom?
Sure. Let's see
Chat.
How poorly I'm going to do this. Okay. Okay, can people see what I hope they can see? We already give the conclusion. Hopefully that's visible. All right, great. All right,
Just so you know, we do see your slides sort of.
yeah, I don't care. It's life. And I can get going. I see.
Yep, sure.
All right, so Will already gave you the where he's going and where things have been gone. And so fortunately, I'm going to start in the beginning of the experiment. An adaptation and divergence in the same system. Noah got married this week, so he's not here, and Bea's at a different conference, so you're stuck with me. So according to Darwin and a whole bunch of other people, they think ancestry matters. The common ancestry is this important thing. So the Darwin's famous notebook drawing and the only illustration in the origin of species, how important the origin, that the ancestry is, presumably for understanding diversity and maybe for evolution itself. A lot of people picked up on this. Haeckel, amazing, one of the first trees that's got any sort of detail on it. Well, not right, but the idea is there. And a relatively recent paper that sort of describing the diversity of life, and you probably can't tell, but eukaryotes are popping out of archaea on this graph. So ancestry seems to be important for our understanding for how diversity arises and the complexity of life. So, well, that's all well and good that we think it matters, but does it really matter? I mean, we have this tree here of the multicellular life. This is not exactly the same one that Will showed, but it's got the same pictures on it. You have all this diversity here. Why do you have all this diversity? Presumably it's because the ancestors you start with matter. But how and why do they matter? That's a lot harder because, as Will said, it's hard to go back in time, even though Batul actually managed to do it, at least in some way, earlier this week. How do you do that experiment? Well, one way to do it is you start out with a unicellular yeast like Saccharomyces cerevisiae. It's one unicellular yeast. You take a bunch of replicates, you evolve them in time with settling selection, and you get multicellular yeast out. And we call them snowflake yeast because Will used to be, and I still am, in Minnesota, where snow is ever present, albeit not quite today. So if you look at those populations, are they the same or different? I mean, they kind of look, they're all the same. They're all snowflakey, but are they the same? So this is where Noah stepped in and said, I want to actually know if they are. So he looked at the distribution of sizes using biomass and number and asked, well, how can I quantitate them? Are they actually different? And so this is a slightly odd graph that shows how similar or not, they're not at all similar. The distributions of sizes of those 10 replicates are quite different. So if you actually looked at the diversity of across populations, it's much greater than the diversity within populations. So you can easily distinguish after just a few weeks of selection, the populations from one another, just on the diversity from single clones from those populations on on how big each one of the individuals is within it. He next he sequenced all the strains that we had, and we had sequenced like a gajillion of them, and re-identified and made sure that we actually had them right, all the mutations that were responsible, made knockouts in those, and then sized them again and pasted on in a genotype phenotype map how much of the diversity that we see among these replicates can be associated with the causal mutation in that we had identified. And so there's basically 5 groups: ACE2, NBA1, NAP, whatever. There's a whole bunch of different mutations that can give you multicellularity. And if you make knockouts just in those genes, double knockouts, because single knockouts won't do it, you end up getting over 50% of the variation in those populations. So it doesn't get it all, but it does get a substantial amount. And that's what this shows here. And there were more, we had a variety of, we had multiple individuals from different populations, because it turns out that multiple individuals from the same population actually didn't always have the same mutation. So you can actually do a genotype phenotype map and understand a substantial amount of the diversity from the initial experiment. That's right. So let's look at ACE2. ACE2 was, is known before we even did this experiment that it will give you something like a snowflake yeast. And if we go to the very first steps of when ACE2 goes, when you get this ACE2 mutation, what happens? Well, it turns out if you have a heterozygote that's ACE2, it's got the ACE2 wild type allele and the derived allele, it's actually smaller than its ancestor. So it's not bigger. So the first step is actually you get small. And then and that smallness makes you settle slower than the wild type. So it moves you phenotypically in the wrong direction, which is not, we didn't expect that. And it sort of tells us that gene dosage is important, and there possibly can be the biochemistry and the gene regulation underlying snowflake is actually really important for telling us how it can evolve, at least how it evolves initially. So if you then take those heterozygotes and you look at how fast they evolve multis from them, so this is 8 passages, so a week-long experiment basically, the heterozygotes evolve multis way faster, which isn't entirely a big surprise, but that's why even though you get small for the first step, being ACE2 heterozygotes gets you faster along to being a multi. So in the very first step, you move in the wrong direction, at least in terms of size and albeit in settling, but not in a big way, but you don't move in the right direction at all. All right. So here's back to the graph that Noah generated with all these different variants, of which I've only talked briefly about ACE2 especially. And if you then take all of these and do what I think my immediate prior speaker said, let's look at transcription. And you ask, well, what do we see? So there's a lot of variation, log phase and post-dioxic growth, which is the important things for yeast. You'll see there's a lot of diversity among the 5 knockouts that we made. They're double knockouts, and tremendous amount of diversity. And in fact, if you look at all of those knockouts, there's only 4 changes in common. And those 4, for people who are involved, aren't surprising ones. If you let it be just any 4 of those 5, you get a slightly larger number of of transcriptional changes that are shared, but it's a very small number. It's a very small number that all give you this multi-phenotype by basically knocking out how cells divide from one another during the beginning of the experiment. So why is this? Why is this relevant for the thing that I at least posited this talk is about, in the beginning part, and why do we have all this diversity? Well, it turns out for this experiment, at least, there's a lot more differences in the transcriptional responses. They're vastly more different from one another in the underlying changes than they are the similarity. They only share 4 or 5 changes really that are transcriptional. The vast majority of them are divergent in this particular design. And I think if we had started the experiment, if one starts a long-term experiment and you don't use ACE2, you might get a different outcome, because it turns out you have a really quite different bug that you're playing with instead of ACE2, because ACE2 and any of the other pairs, they only share a very small fraction. So here you can see it, the differences already, quite a lot. So what I would argue that the unselected differences, the ones that aren't important, are actually more important for giving you divergences in a variety of environments. And I think I finished on time. Thanks.
You finished with plenty of time for questions.
Wow, as if I planned it.
I think there's one in the chat from
I have to look at my
Ratcliff.
chat window.
Let's see, what's the intuition behind ACE2 / all caps ACE2 reducing size? What is the size range of size classes being lost in the HET?
Right. So it turns out that Y55 makes short chains of cells, unlike many other cerevisiae. And being a heterozygote, one reduces that and actually makes the individual sizes of the cells smaller. And as much as I'd like to say that, oh, well, I know exactly enough yeast biochemistry to tell you why being a heterozygote for ACE2 would do that, I'm not going to spin that tale today. It is a big surprise. It is the thing that's unique about ACE2, and actually this is probably true for the other ones. ACE2 is really the only transcriptional activator of all the other ones. The other ones, they only indirectly, by means that aren't entirely certain, affect those genes. They're not transacting regulators. So it's a bit of a surprise if you're a yeast geneticist that you have these indirect regulators that can give you this. And I think I have to get out of sharing if I'm going to be able to see the chat window, or no, I'm not. I can see it right there. I'm just not used to that. All right. Okay, is that the only question? I'm not sure. If so, no worries.
Mike.
Yep.
It's Jay. Have you thought about maybe doing this with a different starting genotype to test that thought?
So I can give 2 answers. One, we've already redone the whole experiment with a different yeast, K. lactis. And if you do it with K. lactis, you get completely different results and exactly the same. In K. lactis, you get only 2 variants. You get ACE2 and AIM44, which is actually in here. ACE2 is by far a dominant, unlike in cerevisiae. And you also, unlike in cerevisiae, you get cooperative settling. Different genotypes co-settle together. So at the biggest difference, starting with completely different ancestor, 100 million years divergent, because that's what they are, they're exactly the same and completely different. Wow. Depending on, they're exactly the same in the genetic mechanisms, they're a subset, and they're completely different in that they take advantage of potential for cooperation among the genotypes to co-settle.
Wow, thanks, that's a great answer.
Yeah, and Jolene K, another grad student, has actually done a bit more on this, building on stuff that Maria Rebolledo Gomez did that shows that there is contingency, and you can show how it, so she's done backward forwards experiments and shown that actually contingency builds on itself, and the lines that start with different ancestors stay different. They stay different in the tempo at which they re-evolve multicellularity after losing it, and they stay different in the forms that they have. So you really see in this experiment, like the LTE, contingency really matters. It just didn't take 50,000 generations to get there.
Awesome.
So there's still technically time for maybe one more question. Maybe I'll share my screen because I'm the next speaker. If there's another question. I think this is the right screen.
Happy to have contributed getting us back on time.
Yes.
Let's talk.
Yes. Let's see.
Yeah,
All right.
well, being from New York-ish.
Can folks see this? We good? Okay. So let's see how good of a job I do at moderating my own time. Someone feel free to independently time me and shout at me if I go over. Okay. So I'll go ahead and get started. So I'm Alex Lalejini. I'm a postdoc at the University of Michigan, and today I'm going to be talking a little bit about some of my work during my PhD, looking at the evolutionary consequences of phenotypic plasticity. And so with many great projects, I had some wonderful collaborators for this work that I want to acknowledge, Austin Ferguson, Dr. Nkrumah Grant, Dr. Charles Ofria. And so let's all get on the same page about adaptive phenotypic plasticity. And so this is a phenotypic plasticity, a mechanism that organisms can use to do the right thing at the right time. So to use sort of a fun example, imagine that you're R2-D2 and you see Ian McGregor in the distance. And maybe the right thing to do is to roll on up and have a lovely chat. Oh, let's see that in a different scenario, you see an angry, scary Darth Vader. He has some unresolved anger issues in the distance. And probably the right thing to do is to quickly scurry away or risk becoming a heap of scrap metal. And so a little bit more formally, phenotypic plasticity is the capacity for an organism to express different phenotypes in different environmental conditions. And so this is the mechanism that E. coli use to survive in the presence or the absence of oxygen by plastically switching their metabolism from aerobic to anaerobic. And so we use experimental digital evolution to study how adaptive plasticity evolves, and then also how the evolution of adaptive plasticity influences subsequent evolutionary dynamics. And so specifically for this work, we use the Avita digital evolution platform, which is this nice open source software system where we have experimental populations of self-replicating computer programs. And so in this talk, I'm going to be specifically focusing on the evolutionary consequences of phenotypic plasticity. And so let's talk a little bit about plasticity in Avita. And to do that, we need to talk about phenotypes. And so in Avita, we're evolving these self-replicating computer programs. And if you plop down one of these self-replicators inside of a digital environment and you let it do its thing for a little while, it will eventually produce some offspring. Now, these digital organisms can also metabolize resources in their environment by performing computational functions. And by doing this, they can improve their speed, the efficiency of self-replication. And often in a lot of experiments, the more different types of logic functions or computational functions that these organisms perform, the faster they're able to produce offspring. And so just what I want you to take away is that in this work, a phenotype is just the set of computational functions that a digital organism performs. And so with Avita, we can control the value of these functions in the environment. So in that example, NAND, this computational function, was beneficial. So if an organism performed it, it improved its replication efficiency. But we can also set up the environment so that NAND is harmful. So if you perform a NAND, it actually degrades your ability to self-replicate. And so with these sorts of controls, we can construct environments where organisms need to do the right thing at the right time. So we can construct, so for this work, we constructed two environments, so environment A and environment B. And for environment A, we set up three computational computational functions to be rewarded, or they were beneficial, and three different ones were punished or harmful. And environment B was sort of the same thing, except the opposite functions were rewarded and punished. And what we can do is we can fluctuate repeatedly between environment A and environment B over and over again, and sort of create this environment where organisms really do need to do the right thing at the right time. So when the environment is in the sort of this orange state, environment A, organisms should really only be performing not, and or, whereas in environment B, organisms should really only be performing NAND, and not, and or not. And so phenotypic plasticity in Avita, at least for this work, is just that a plastic digital organism is one that performs different computational functions in different environments. So in this case, environment A and environment B. And adaptive plasticity is just doing the right functions in the right environment. And so to investigate the evolutionary consequences of phenotypic plasticity, we set up this two-phase experiment where we seed populations with that common, that default self-replicator capable of no functions or regulation or anything like that. And we evolve those, these populations for about approximately 30,000 generations in a fluctuating environment, fluctuating between A and B. And we're calling this the preconditioning phase. So this is just giving populations a chance to adapt to this fluctuating environment. And then what we do at the end of that approximately 30,000 generations is we take a sample and we transfer it over to make new populations with the samples, and do the same exact thing for another about 30,000 generations, except during this phase, we actually track the evolutionary histories of the populations. And basically what this whole two-phase rigmarole lets us do is exclude early adaptation dynamics from our analyses. And so for this work, we had sort of really simple setup. We had three treatments. We had a plastic treatment where we allowed phenotypic plasticity to evolve. We had a non-plastic treatment where we actually disallowed phenotypic plasticity from evolving. And then we also had the static control where we evolved populations just in a static environment the whole time, where we rewarded all of the computational functions. And so using those evolutionary histories, we can ask a couple of questions. So first, we wanted to know, does the evolution of adaptive plasticity sort of influence the speed of subsequent evolutionary change? We also wanted to look at whether or not adaptive plasticity promoted or constrained the evolution of novel traits. And then finally, does the evolution of plasticity influence the propensity for deleterious alleles to accumulate? And I don't have time to go through data for all of those things, so I'm just going to talk about the first one, which unfortunately maybe is the most boring, but if you're interested in the other two, I'm happy to chat about it either offline during the question session, or you can check out, I think we have a preprint of this work on bioRxiv that I can link to. Okay, so why might we expect phenotypic plasticity to have anything to do with the speed of evolutionary change? So why might we expect it to increase the speed of evolutionary change? Let's just step through this example plastic genome. So keep in mind that to digital evolution, we're evolving these computer programs. So each one of these little rectangles represents an instruction on a plastic genome. And the color of the rectangle is indicating which set of environments that instruction is expressed in. So all the yellow rectangles are saying that that instruction is expressed in both environment A and environment B. The purple are saying it's expressed in environment B, and the orange are saying it's expressed in environment A. And basically, the point I want to make is that not all regions of the genome are expressed in all of the environments. And so when this plastic genome is executing in environment A, you've got this grayed out region of the genome that is unexpressed. And so mutations can accumulate there. And then when the environment changes to environment B, those mutations might be revealed, sort of process as cryptic variation. And sort of this process of potential increased rate of mutation accumulation in plastic genomes could lead to increased rates of evolutionary change. Little caveat, in this example, I've just shown like one genome here. As I click through, just this process of mutation accumulation occurs over multiple generations, not in one generation. Okay, so that's sort of maybe like a mechanism behind why we might expect the evolution of plasticity to increase evolutionary rate of evolutionary change. What about decreasing the rate of evolutionary change? So if we think about this fluctuating environment between A, B, A, B, A, B, non-plastic populations sort of need to keep evolving, relying on de novo mutations to continuously adapt to these environmental changes. Whereas the populations of plastic organisms can sort of just regulate their phenotype and do the right thing at the right time. So in this way, we might expect to see non-plastic populations evolve more rapidly because they have to in order to keep up with changes in the environment. Okay, so to sort of get at this question, in that experiment that we performed, we looked at 3 sort of different lenses on evolutionary change. We looked at mutation accumulation along successful lineages, accumulated phenotypic changes along successful lineages, and then we also looked at the frequency of selective sweeps. And so in these graphs, basically I'm going to show you mutation accumulation on the left, phenotypic changes on the right. On the y-axis for both of the graphs, as you go up, we're increasing the amount of evolutionary change, and then the static treatment's on the left, non-plastic treatment is in the middle, and then plastic is on the right. And so when we look at the data, we see a pretty clear signal that in the non-plastic lineages, they're accumulating many more mutations, and we're seeing many more phenotypic changes along those lineages than in the static or in the plastic lineages. And just note that this is on a log scale, so even this like tiny distance is actually pretty huge. And the other sort of interesting takeaway here is that these plastic lineages actually look a whole lot more like the static lineages than they do the non-plastic lineages, despite those 2 treatments having identical environments. And so, so big takeaway, non-plastic lineages exhibited more total evolutionary change. And so next up, just going to take a look at the rate of coalescence events. So as sort of here, we're looking, as you go down on the y-axis, we're increasing the speed of evolutionary change. And so when we look at the data, we sort of see basically the same story as the other data, where non-plastic populations are undergoing selective sweeps much more frequently than the static or the plastic populations. And so the takeaway here, just non-plastic populations undergo much more rapid evolutionary change. And so sort of start to wrap up. So why are these non-plastic populations evolving more rapidly? So we dug more into this with some data that's in the paper. Basically, the non-plastic populations are relying on mutations to repeatedly align their phenotype with the fluctuating environment. And the plastic organisms can just sort of sense their environment, adjust what tasks they're performing. So in a sense, the plasticity makes the environment look like a static environment. And so overall takeaways from this work, so the evolution of adaptive plasticity stabilized populations against environmental fluctuations. I showed you that this stabilizing effect sort of resulted in slower overall evolutionary change. Things that I didn't show you, but I wanted to point out, the sort of stabilizing effect of plasticity also improved the retention of novel traits, and then also resulted in the accumulation of fewer deleterious alleles. And then overall, sort of across the board, across all the experiments we did, the dynamics of the adaptively plastic population sort of resembled that of the populations evolving in a static environment, more so than they resembled the dynamics of non-plastic populations evolving in sort of an identical fluctuating environment, which was neat. With that, I will take any questions. And cool, I stayed on time. Let's see. All right. I see question in the chat, maybe from Ben. I wonder whether it will be the case that the evolution of adaptive plasticity facilitates the evolution of new forms of plasticity because the same plastic machinery can be redeployed. Does plasticity beget further plasticity? I think that's an awesome question. And that's definitely a sort of next step that we're interested in. And I might be wrong, but I think Austin Ferguson is planning on maybe looking into that a little bit. In Avita, it's a little weird because the plastic machinery there is really specific. And it's specific in the sense that the way that Avita digital organisms regulate their phenotype or their functions are with these like if statements. For the most part, there are other ways that they can regulate them, but the most common way that I've seen is with if statements that basically toggle a single instruction on and off. So like in Avita, it may not necessarily be the case that like plasticity at one level begets more plasticity at the next level, because the plasticity at the bottommost level is so incredibly specific. And that actually is a thing that we talk about in the paper, because it's probably why we don't see much evidence for cryptic variation accumulating in these experiments. So hopefully that answered your question. And I think that's probably all the time that I have. I'll take the rest of the questions in the chat if there are more. Let's see, let me stop sharing my screen. And let's see, Rohan, are you available to share your screen?
Yeah. Okay, can everyone see my screen?
Yep.
All right, great. I will just get started. All right, so I'm going to talk about purifying selection in Rich's long-term evolution experiment in coli. I'm very excited to kind of talk about my lockdown science. So I'm going to talk about like really several kind of like short stories. So everything here is available online. So if I'm going too fast or you miss something, please ask questions or email me, or you can also just look up the actual papers or the bioarchive stuff. All right, so this work is an outgrowth of like a very fruitful collaboration with Increment Grant, where we were basically just like, poking at Rich's long-term evolution experiments, like the published data, and kind of like thinking about how we can draw like interesting new conclusions and interesting new science using pre-existing data. So some of this early work is already published in Chromospecies, which is this particular chapter is published in American Naturalist. If you haven't read it, you should check it out. It's a great paper. So I just want to give a shout out there. It's rich as well. All right. So since I'm talking about purifying selection, something that's kind of interesting is that sometimes what's not in the data is actually quite important. So what does this mean? So I'm going to kind of give like a statistical story that some of you may have heard before. So during World War II, a statistician named Abraham Wald, who's now very famous, was doing studies for the U.S. Army, during World War II. And so what he was particularly studying was looking at damage on like American bombers, trying to figure out like how can we, save lives, get like more pilots back, more planes back, right, without getting shot down by the Germans. And so he was like analyzing like these aircraft which had like bullet holes. The question is, how can we better defend the aircraft? And so the Army's conclusion was, well, we need to put armor on all these spots where we see all the bullet holes. Like wherever we see the data, that's showing the parts that need to be protected. And Abraham's great insight was, no, this is exactly the wrong conclusion, because what we're really seeing is that the places where we don't see the bullet holes, those are the planes that don't make it back. So these are the things which are missing in the data. So here, the really important part are the things which aren't getting hit by bullets, right? Because when they get hit by bullets, like we don't see those planes anymore, right? So essentially what I'm doing in this work is applying this kind of thinking, but to like long-term evolution experiments. Rather than looking at where the mutations are, which is what we do most of the time when looking for positive selection, now with like, 60,000, now 75,000 generations of data, we can start to look at where the mutations are not. And see what conclusions we can draw from that. All right, so the reason why we can do this is because half of the long-term line populations have evolved a very, very high mutation rate. And these dynamics are very interesting. There's decades of work on this. And Kurma and I wrote kind of a recent paper reporting kind of like cool patterns that we see in these data last year. If you're interested, you should check that out. But really the main point I want you to just take away from here is that half the long-term populations have like a very high point mutation rate. We call these like mutators or hypermutators. So in these populations, we can kind of get an idea of like where we don't see mutations. And that gives us very valuable information. All right, so one hypothesis that I kind of came up with, like, well, in my dissertation, I spent a lot of time kind of like thinking about how we can compare Rich's experiment to patterns of variation in nature. So going along with that kind of line of thinking, I did that kind of follow. So it's a very well-known pattern when you look at pretty much genomes across the entire tree of life, which is, you guys see my pointer? All right, so people know that protein abundance in general anti-correlates with evolution rates across the tree of life. So what that means is that when you have these very abundant proteins, these can evolve quite slowly compared to proteins which are like less. And so like one kind of inference from that empirical fact is that purifying selection causes highly abundant proteins to evolve slowly. The causes of this are really debated. People are really arguing about this. People really don't understand the causes of this. Like people talk about mistranslation, protein folding, things like that. But there really isn't any consensus in the field for why that's true. So if we assume that this hypothesis is true, if we just assume that purifying selection is what's causing highly abundant proteins to evolve slowly, then we should be able to, eventually predict that protein abundance should anti-correlate with the density of mutations per gene across hypermutator long-term line populations. And so previous studies was, essentially like look at evolution trees across, millions or billions of years of evolution, and then make like an anti-correlation between, protein abundance in the lab and evolution rates in those kind of like phylogenetic data. Here I'm just like counting mutations in Rich's experiment. I'm just saying like, let's count mutations per gene, and we'll assume that, really important genes are not going to get hit by mutations so often. So here it's sort of like mutations are kind of like the bullets, and we're looking for genes which aren't really getting hit by bullets as often as we would expect, because you're so what I did was I just like looked at published data from a particular study. I think this is from like UC Austin, and I think Chris Mokes is on the study too. Essentially, it's like some like genomics. It's some RNA-seq and proteomics data for REL606 run in I think GM250. And then so what I did was I took like these time course data of like abundance of RNA and proteins, and I just kind of like put like a line to the density of observed mutations per gene. And what I find is a very strong, a very significant anti-correlation between mutations and how abundant proteins are. So this is very consistent with what's seen in kind of like natural data. What's really surprising is that, in fact, we see the opposite correlation when we look at synonymous mutations. And so often synonymous mutations as like a kind of like a really rough proxy, it's often a very rough proxy for neutral evolution. And so this really seems to indicate that it's purifying selection which is driving this anti-correlation, not some kind of like mutational dynamics or something like that. So this is something that the reviewers actually asked me to do, and this was like a really cool pattern which I didn't even expect. But it turns out that when I kind of like look at these data over time, kind of like subset the data, look at the first 10,000 generations and 20,000 generations and so forth, the anti-correlation gets stronger over time, but for synonymous mutations, it stays positive over time. So this really seems to indicate that there's something specific about, mutations which are not synonymous. And so this is kind of like additional evidence that purifying selection is kind of driving this trend, and this is consistent with what we see in nature. So this is nice kind of like convergence between natural patterns of molecular evolution and what we see in the lab. All right, so this kind of like one tale I'm going to tell you. I'm going to tell you a bit about another tale about purifying selection, which is a very kind of different framework. So here it turns out that I found some evidence that the protein-protein network shows evidence of purifying selection in Rich's experiments. And so the genesis of this was that I read this kind of interesting paper that at first I didn't really agree with. I was very skeptical. That's a better way of putting it. Where these authors who are very well regarded were looking at like network evolution across the tree of life, and so they came up with some metrics for looking at a protein-protein interaction network, and they had this idea of protein network resilience. So the idea of resilience is that you kind of like knock out nodes in the network, you kind of ask like how quickly does that network break down? And so a resilient network is something that you can kind of like remove a lot of nodes in that network, and it kind of stays connected. And so a really fragile network, you remove a few nodes and then it falls apart really quickly. And so they kind of came up with some measurements for how to kind of characterize network resilience. And the idea is that you kind of like randomly remove nodes and you kind of measure like how quickly does it fall apart. There's often a particular threshold in which the whole network becomes disconnected. This is just like very interesting kind of like math that people do. So kind of like took the math, the equations from this paper and applied it to the genomic data in Rich's experiment, because now we know like we have very good understanding of the E. coli protein-protein interaction network, and we know kind of like what genes in Rich's experiment are kind of like affected by mutations that kind of like break proteins. So mutations which are removing nodes from the protein-protein interaction network. So essentially, I'm just asking, is this notion of resilience at all relevant to the protein-protein interaction network that evolved in the long-term experiment? So essentially, the idea is that, imagine we have an ancestral protein-protein interaction network. Over time, mutations are kind of like removing those nodes. These red dots kind of represent maybe genes which are essential that you can't take out. And so I tried two different randomization procedures. One in which you can remove any protein. So here, I remove like this, essential protein. And then here, I say I cannot remove the red nodes because that would lead to like an organism that can't possibly survive in the world, right? So I tried a couple different procedures. One where I can remove any protein, and another where I only remove those proteins which actually were removed in Rich's experiment. So anything that was not removed, I don't really mess with. What I find is that in both cases. So here, this red line is the actual data. So I'm kind of plotting the change in resilience over time. So here, the red line is like the actual change. And then the yellow line is just randomizing the entire network. And then the blue is just removing genes which are removed in the long-term line. And for both of these cases, I always find that the slope for the red line is in general higher than the yellow line. It's a very strong pattern. And to a lesser degree, it's higher than the blue line. So this is quite significant. So one thing that I thought about was like maybe it's just that we can't remove essential genes. But it turns out, and again, something that reviewers kind of pushed me to do, it turns out that essential genes don't seem to be enough to kind of explain this result. And that's because if I take like the ancestral network and remove essential genes, it turns out that on average, it actually increases network resilience, which is the exact opposite of what I expected. And so it seems that really what's going on is that there's some kind of purifying selection on like the topology of the protein-protein interaction network, such that when you remove certain proteins, that means there's a stronger purifying selection on what's left. And so you don't want to remove what's left because then you're actually causing problems with like the protein-protein interaction network. So that's like it's kind of like second story. I think I'm probably running a little bit short on time. But very, very quickly, I want to talk a bit about a method that Nkrumah and I developed. Okay, let's check it. To look at purifying as well as positive selection on gene modules. And so we have this preprint called STIMS that, hold on a second, I'm trying to get the next slide. Okay, here it is. Unfortunately, I don't really have a lot of time to explain maybe, but you can check out our preprints. The idea is that we essentially come up like this bootstrapping method. This method is also used in the MMAT paper, so you can also check that out as well for description of the method. Essentially, we look for like groups of genes where there are fewer observed mutations in those genes than you'd expect by doing like a randomization test. And using this kind of method, we can kind of like find like a group of genes that we're interested in and ask whether or not it's enriched or depleted at mutation. And so very briefly, I've just been kind of like using this on account of a lot of different kinds of genes. And we find some like interesting patterns. So here is some like interesting kind of like unpublished stuff where I was applying some metabolic genes. And in some cases, it turns out that some particular modules of like essential metabolic genes show like very strong evidence of purifying selection in some populations, but not others. And so this is like evidence of like kind of idiosyncratic purifying selection in say in R-1 that we don't really see in any of the other populations. And so it's really a mystery why these patterns are arising. And so I think it's quite interesting that we can use the molecular data to kind of get information about kind of like either parallel evolution or idiosyncratic evolution in like the experiments, and we can kind of like maybe generate hypotheses for like what's going on and what's generating like these idiosyncratic evolution paths for like future work. And of course, with like 75,000 generations, now we have like even more data to look at like purifying selection. So that's pretty much it. I want to thank Beacon and my collaborators for helping with all this work. And any questions? I'm sure there's quite stuff in the chat. Okay. Yeah. Does anyone have any particular questions? I think I'm over time, unfortunately.
You're good on time, so we have time for maybe one or two questions.
I have a question, Rohan, and it's sort of in light of John's his talk on the gene expression stuff. In the interactome, sort of the protein-protein interactions, what would a gene like NADR look like in that network? Because it's it's interacting via regulation with all those genes that John showed were upregulated. So anyhow, it'd be interesting to try to tease out how much of this is sort of regulatory effects and how much is metabolic. Different kinds of interactions, I guess.
Yeah, that's right. I think I can take a look at those data sets. So I tried this method on 2 different data sets, kind of like for robustness. I believe the mixed data set that I look at is it's from the string database. So that actually may include some regulatory interactions because it kind of like incorporates like many different kinds of evidence. I'd have to kind of like look back at that to see whether it includes regulatory interactions or not. The other data set, the Chrome data set, that's very specific to actual just like physical interactions. But yeah, I think presumably we actually could look at those differences by kind of like considering the kinds of evidence and have the rules for drawing edges between them. Yeah, so I think we could actually figure that out.
Let's see, and actually let's take the rest of the questions on the chat and move on to Matthew's talk.
Great. Are my slides showing up and my audio is coming through?
Yep.
Great. All right. My Zoom is being a little bit laggy. It's occasionally showing me like a Zoom is freezing up message. So if I cut out, I apologize for that, but hopefully I'll be able to make it through here by the seat of my pants. All right. My name is Matthew. I'm a graduate student working with Dr. Charles Ofria at Michigan State University. I'll be presenting work that we did looking at a case study from a digital evolution system designed to study digital multicellularity that we also did in conjunction with an undergraduate research assistant of ours, Santiago. Come on, slide. Okay. This work is really fundamentally motivated by questions about fraternal transitions in individuality. These are events where kin unite to form a new self-replicating group. These include characteristic examples from biology include evolution of multicellularity and evolution of eusociality. And we're really interested in these kinds of events because of kind of the theoretical interest in kind of how they help to explain the diversity and complexity of life on Earth. So before we get into the case study, I'll bring you up to speed to set the stage a little bit with the model system that we used. Like many other digital evolution models, this model tracks digital cells controlled by evolving genetic programs that occupy tiles on a toroidal grid. In this case, in this system, cells are restricted to interacting only with their direct neighbors. And a really key mechanism in this system is this idea of kin grouping. You can think of this kind of like cells being able to retain some kind of attachment to daughter cells when they reproduce in order to grow over time a multicellular body. So here we've just got one cell with kin grouping color-coded in this visualization. The kin grouping is shown by color, and it'll be the same way in many other visualizations. Cells can, are people still able to hear me? Zoom is being a little bit laggy.
Yeah, you're coming through fine.
Yep.
Okay, great. All right. They can produce a daughter cell and place it in another kin group here, shown by color, starting this new purple kin group. These that can also place a daughter cell and register it as part of its own kin group to grow its group. And this repeats over time, and ultimately these cells are competing for limited space on this fixed size grid. We throw in some functional consequences to this kin grouping system, kind of preferentially selecting for groups that are medium sized by rewarding them with resource, and degrading groups over time to ensure turnover so that new groups have a chance to get established. And in previous work, we've shown that this is sufficient to select for traits that are characteristic of multicellularity, like reproductive division of labor, resource sharing, and apoptosis. So just a little bit more background about where the data that we actually used for this case study was drawn from. We did evolution experiments with 120 by 120 toroidal grids, so about 14,000 tiles. So we started out with randomly generated ancestors and ran them forward for a 3-hour window of evolution that we call a stint. And we repeated this process, kind of saving the population to file and loading it back up into a fresh environment. Repeated this 100 times. So we have 100 stints of evolution, where each stint is kind of a checkpoint along the way. With 4 threads, this was sufficient to observe about 20,000 cell generations. We repeated this process 40 times and selected our case study from one of these replicates. So getting into the case study itself. One of the first things we did once we selected the lineage we wanted to do our case study on was actually do qualitative assessment of the different morphologies, multicellular morphologies that we saw evolve. And so we came up with 10 qualitative categorizations for different kind of multicellular morphologies that we saw along the lineage of this case study, assigned these letter codes. And then we actually went through all 100 stints and manually looked at representative genomes and kind of put them in these qualitative morphological categories. So these images that you see here next to kind of the letters are the different shapes of, you can think of it like multicellular bodies that are evolving in the system. And so I'll walk you through kind of some highlights from this lineage. Starting at stint 0, we saw if this video will play, we saw a unicellular morphology where kin groups were just size 1. Each cell was kind of out for itself. Around, we can go to the next slide here, around stint 14, we saw cells start to form kind of these regularly sized blob kin groups, which we refer to as morph D. Later on, around stint 15, we observed the emergence of kind of these really striking horizontally elongated kin groupings, where these cells were preferentially growing their groups out along the horizontal axis. We refer to this as morph E. And then at stint 45, we observed kind of a secondary novelty where later on in their life history, as you'll see in a moment here, these kind of cigar-shaped groups engage in a burst of secondary growth along the vertical axis, which just occurred right there in this video. And so seeing these kind of qualitative novelties along our lineage, we became really curious about how these evolutionary changes related to adaptation and complexity. And so the first thing we set out to measure was changes in fitness and how they were related to these phenotypic novelties. So I have some pocket slides about how we actually measured this, happy to answer questions, but suffice it to say on the y-axis here, I have
real quick, Matthew, I think your slides are out of sync with your voice, at least for me. I don't know if that's true for anyone else.
Same.
I think I'm just lagging like really bad. Can you see a dot plot now?
Right now we were on stent 45, I think. It might have been the YouTube videos that just tanked it. Oh wait.
I don't think Matthew's in the call.
We don't
he's
have
back.
him here anymore.
He's back.
Okay.
I just got booted, but I'm back. Hopefully Zoom has worked out whatever issues it was having. It's being a lot more responsive now. So, dang, were you all able to see those videos at all?
We saw the first couple, I think.
I think they
Okay.
mostly eventually played. They just weren't lined up with your explanations.
Okay,
of them.
well, I trust you all to be able to kind of do the reconstructive work to put together that, but hopefully this is showing up correctly now. I've got a dot plot up.
Yeah.
So on the y-axis, we have stint to stint fitness change. And on the x-axis, we have evolutionary time going from stint 0, the very beginning with the randomly generated ancestor, up to kind of 100 stints of evolution later. And the color coding and the lettering refer to these qualitative morphological categorizations we came up with. So we can see kind of where morphs D and E first emerged here. Those were the kind of the first regularly sized kind of blob-like kin groups, and then those horizontally cigar-shaped kin group morphologies. And then later on, where we see the emergence of that secondary novelty, where later in their life history, these groups were engaging a burst of secondary growth. What was really interesting about this data was the stint to stint fitness change that we observed that during these novelties wasn't noticeably different compared to kind of other fitness changes that weren't associated with novelty, which was kind of a surprising outcome. And we're still working on kind of tightening up our adaptation assay. But if this kind of ends up holding, that would be kind of a really surprising to be able to see these really striking changes in morphology that aren't necessarily more adaptive than other just adaptive change that's not associated with novelty. We also wanted to try to quantify the complexity of these genomes as they evolved. This first complexity measure kind of looks at a genome like a Jenga tower. And in this measure, we're trying to count up how many different Jenga blocks, when we pull them out, the tower collapses. So if a genome site is like a Jenga block and the tower collapsing is like having a deleterious outcome, we're trying to answer how many individual sites in this genome are critical for fitness. If we take it out, fitness goes down. Pocket slides again about how we actually measured this. Happy to answer questions, but I've got another one of these dot plots up. Again, evolutionary time on the x-axis. On the y-axis, we have this sequence complexity measure. And then the color coding and the lettering are the different morphologies. Some features here that we saw in this visualization. We saw early on kind of a discontinuity in sequence complexity, a big jump in sequence complexity that wasn't associated with qualitative morphological change. Before and after, we categorize it as morphology B. We also saw large changes in sequence complexity that were associated with morphological change. Interestingly, going from morph D to morph E, we saw a big dip in sequence complexity, which we'll kind of return to later. And we also saw periods of more gradual sequence complexity growth. I'm going to kind of speed through this last part here because I think I'm a little bit short on time because of the technical issues. But we also tried to complement the sequence complexity measure by trying to also have another measure that quantifies the richness of interactions that cells are having with their environment. So there's 3 different ways that cells can interact with their environment. And we basically tried to quantify like how many different distinct ways are cells interacting with their environment that are necessary for fitness. Like how many things is DJ genome here doing that's actually important. And so pocket slides again about how we measured these. But really the interesting feature here is going from morph D to morph E, where we first saw that horizontally elongated phenotype. We see a near tripling of interface complexity. But what's really interesting is that's the opposite signal of what we were getting from our sequence complexity measure. And I'll kind of return to that idea again in the conclusion. So to wrap up here, because this is a case study, The takeaways are kind of anecdotal in nature. However, we did see a kind of a loose coupling between novelty, complexity, and adaptation in this system, where although novelty was sometimes adaptive, innovations didn't yield exceptional fitness increases. And at times, even within these measures, within the complexity measure, we saw kind of opposite signals, which I think really underscores the importance of interpreting complexity as kind of a multidimensional phenomenon. I'll skip over future work in interest of time, but I just want to acknowledge my co-authors and Catherine Perry, an undergraduate in our lab, who contributed code to this project. And with that, I'll take questions. Thank you.
Thanks, Matthew. Let's see, I think there was at least one question in the chat from Will Ratcliff. How non-transitive are these interactions? Do later evolved strategies do well against earlier ones, or is it all about just beating the last dominant competitor?
That's a really interesting question. We've definitely seen, I don't have data that kind of quantifies that directly, but we've definitely seen in this environment that fitness is very non-transitive, that in kind of previous work, I did a screen to look for from a bunch of genomes to see if there were cycles where rock, paper, scissors, kind of A beats B beats C, and those definitely kind of show up in this environment. And so I think there's definitely a really strong biotic selection factor here, which I think is something that is a really interesting characteristic of the system, and something we're interested in digging into more.
Let's see, maybe, okay, yeah, one more question from Mike Travisano, and then maybe we wrap it up after that.
So how much exploring of a metapopulation model are you interested in pursuing?
I'm not familiar with that terminology. Could you elucidate a little bit more
So
about
you have
metapopulation models?
So you could have migration among populations, and that would give you some power to investigate the sort of non-transitivity.
I see.
And it could be illuminating to sort of give you ideas about who wins given different migration schemes and different local biotic interactions.
Yeah, that's definitely a direction we're really interested in going. We haven't done work in that direction yet, but one really interesting thing I'd like to do in the future is investigate how having kind of subpopulations with migration, which you would expect to kind of preserve diversity and be putting genomes into situations where they're having to interact with new neighbors, how that might relate to the evolution of complexity, and perhaps through some kind of weird stepping stones adaptation. And we're also interested in potentially trying to build in systems that promote long-term coexistence and seeing if that relates to kind of adaptation and complexity. So I think that's something we're really excited to explore. Yeah. Great, I will end my share here. Thank you again, everybody, for your patience with the tech issues.
You handled it wonderfully, Matthew. as best as anyone can. All right, I think thanks everyone for a wonderful session. I'm gonna stop the recording. Mike Weiser, my tech support, would you mind exporting the chat?
Yep, I'll be able to do that.
Okay.
Also, I just want to jump in and thank everyone for coming to Beacon Congress. This is the end of the last regular session. I think various folks are still going to hang out on GatherTown if anyone wants to head over there, but otherwise, great to see you all. And thank you for all the great talks.